Hello everyone. Today we will see a video on multiple sequence alignment algorithm. We'll have a quick tour on the algorithms, various algorithms used for multiple sequence alignment. Multiple sequence alignment is a key technique used for aligning three or more biological sequence to identify conserved region. It is a comparative genomic, phylogenetic and functional annotation tool. There are three major algorithmic approach used. They are progressive alignment, iterative alignment and consistency based alignment. Let's see the various algorithms one by one. So under progressive alignment we have popular tools like ClustalW, MAFFT. So they build alignments in a stepwise manner. Then you have iterative refinement algorithms like muzzle, which are popularly used, and you also have algorithms which gives high consistency like tea coffee. Here they use sequence analysis approach like global and local alignment for improved accuracy. Now in the coming slide, let's see about each and every algorithm in detail. So progressive alignment approach is here you see a flowchart explanation of the progressive alignments. Initially the input sequence are unaligned. So the first step usually the process starts with multiple biological sequences that need to be aligned. The sequences could be DNA or RNA or protein sequences. First we will be doing what is called as building a distance matrix which computes the pairwise alignment. So this distance matrix usually can be constructed using scoring matrices like Blossom and PAM. Once this is done, you will compute the similarity scores between the sequences pairwise. Based on the pairwise is done for all the pairs of sequences in our set. Once this step is done, we will go about constructing what is called as a guide tree. A guide tree is nothing but a phylogenetic tree generated based on pairwise similarity searches. Either a neighbor joining approach or UPGMA, which is unweighted pair group method, and arithmetic mean method is used for constructing the tree. Once the guide tree is constructed, we would go about doing what is called as a progressive alignment approach using the guide tree. So the algorithm aligns sequences in order determined by the guide tree, and it starts by aligning the most closely related sequence first and then progressively adds others. So once this step is done, we will generate the multiple sequence alignment. So during these steps, gaps are inserted to optimize alignment based on evolutionary relationship. Post this step, a refinement step is done. Some tool do it, some tool doesn't do it. So iterative method to correct potential errors is introduced during the progressive alignment. So if you happen to see the pros of this alignment, which is very computationally efficient and widely used by ClustalW and MAFFT methods. Cons usually are early alignment errors propagate and affect the final MSA. They are widely used in construction of phylogenetic tree, motif construction and evolutionary analysis. Now let's see a more refined method called as iterative refinement method. So here are the steps for iterative refinement method. Initially, a group of unaligned sequences are started. There could be either DNA, RNA or protein that needs to be aligned is taken. Then we generate what is a distance matrix. Okay, so this is an initial alignment that is usually followed in the previous alignment method, like using progressive alignment. This starts as the starting point for the refinement. So this is the first refinement that is done. Then once this refinement is done, we are introducing a alignment approach here where a split alignment into subgroups is done. So based on the alignment is further divided into subgroups and newer alignment subgroups are generated. So this MSA is broken into subgroup by removing one or more sequences from the alignment. And this step allows the algorithm to realign sequences independently. Once this step is done, we realign the subgroup independently and removed sequences are realigned to reset for MSA. This process is repeated iteratively to optimize the alignment. So you can see this is iterative alignment happening. Once this is done, uh, the scoring function like sum of pairs method or weighted sum of pairs method or consistency score is used to evaluate the quality of the alignment. If the score improves, the changes are accepted, otherwise they are discarded. And then this iterative steps happens till the convergence happens. This step is repeated multiple times till the point the alignment stabilizes, and no meaningful improvement is observed in the multiple sequence alignment. So the best alignment is got as the final MSA as the output. So the key points to take in this or pros would be improve accuracy by correcting errors in the initial alignment. Cons would be computationally expensive, especially for large data sets. Muscle and MFAT uses these kind of methods. They are usually used for protein structure prediction, phylogenetic tree reconstruction, and functional genomics. So one more algorithmic approach is present called as consistency based, where it is combination of primary local alignment methods and global alignment methods. Let's see about them in detail. So here again the algorithm starts with multiple sequence alignments, and then the sequence are usually different species or different families required for careful alignment, and a pairwise alignment is computed based on blossom and pair matrices, and they are generating based on scoring alignment and likelihood or specific alignment. These pairwise alignment are generated. So you have pairwise alignment generated using local alignment method, and you have pairwise alignment generated using global alignment method. So based on these alignments, we would be constructing what is called as an initial alignment library. So an alignment library of all possible alignment is created. These alignments are stored as sub-alignments that are later guide the full alignment or MSA generation. So based on this, primary library is created, and this primary library is used for consistency. So how do we access the consistency across alignment? So the algorithm ensures that the pairwise alignment agree with each other. For example, if a sequence A aligns with sequence B and sequence B aligns with sequence D, then A should be aligning with consistently with sequence C. Based on this consistency information, an MSA is generated, which is called as which is done by the progressive alignment, and the algorithm checks for multiple sources for the agreement. Like you have the extension and extended library, and then the local and global alignment. The algorithm checks all this for the consistency. Once this step is done, a refinement or optimization happens. The alignment is refined iteratively to maximize consistency and accuracy. The algorithm reviews inconsistency and maximizes gaps and enhances sequence conservation. Then you end up having the final MSA output. So this output is more accurate than the traditional progressive or other approaches that we saw. This is particularly useful for sequence with complex evolutionary relationship, usually with the eukaryotic systems. So key insights would be why we use consistency-based MSA. Unlike progressive alignment, which is dependent on single guide tree, this method ensures consistency across multiple sequences alignment. It is less prone to early stage alignment error. Key tools like tea coffee uses the consistency based method. So the application involves functional and structural genomics, evolutionary biology, and protein secondary structure prediction. Now let's see a small example of what happens to the alignment upon using this method. So you have three sequences. Now just after doing MSA, upon introducing gaps at the right region, the alignment would improve. So these are few data formats that MSA uses, like FASTA, Clustal, PhyLib, Stockholm, which is dependent on Hayden-Markov model, and then Nexus for evolutionary studies. Key applications of MSA include include identifying conserved motifs and domains, constructing phylogenetic tree, predicting protein structure, drug discovery, and biomarker identification. So here is a quick tour on various multiple sequence alignment algorithm. Thank you for your patience listening. Thank you.